Hello everyone, and welcome to today's live broadcast, Exploring Pharmacological Interactions with Matrix, the Innovations and Challenges of NCATS Drug Combinations /HTS platform, presented by Dr. Kelli Wilson, research scientist in the Information Technology Research Branch at the National Center for Advancing Translational Science, NCATS, at the NIH. I am Marjorie Torres of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want, at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window, and type your questions in the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Kelli Wilson. I will now turn the presentation over to her.
Thanks, Tiffany, for that introduction. And so I'm going to get started now with my talk, which is entitled Exploring Pharmacological Interactions with Matrix, the Innovations and Challenges of NCATS Drug Combination /HTS platform. So the talk that I'll be giving today is just a brief outline. So I'll just give you a brief introduction to NCATS, the institute at the NIH I work at, for those that don't know about it. Then I'll talk about reasons why you may want to implement combination high-throughput screening, or cHTS as I'll refer to it today, and the requirements that you would need to implement this type of platform in your own institution. Then I'll talk some about acoustic dispensing challenges and solutions. I'll also talk about our screening workflow and some considerations you'll need before starting, and also some of the challenges and solutions we have developed here for data analysis. And lastly, I'll give a couple of examples of scientific questions where you can apply drug combination screening to really understand drug basic biology, or to try and explore drug repurposing for various disease types. And then that will be the end of the talk. So let's get started. First, you may be wondering, what is NCATS? So NCATS is one of the newer NIH institutes, and it stands for the National Center for Advancing Translational Science. And the mission of NCATS is to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. And today, the talk that I'll be giving is really focused on the innovative methods and technologies development that we've done here. And so NCATS basically is structured as part of multiple groups. This is just your typical pharma pipeline, where you start with a target for your particular disease, and then from there you move down this line towards hopefully FDA approval. And so the group that I'll talk to you about today is really focused on working in the space around assay development, probe lead development, and lead optimization, leading up to preclinical development. And so hopefully today you'll learn a little bit about what we do at NCATS, and we're a highly collaborative institute. Even though we have lots of groups within the NCATS umbrella, all of these groups are collaborative. We have a stem cell technology facility. We also have an RNAi screening group. So there are many, many opportunities for collaboration within NCATS. And so if anyone is interested in learning more about that, please feel free to contact me about that, and I can try to direct you to the proper group. So then let's move on to why would you want to use this combination drug screening platform. And the goal for our program has been to look for novel drug synergies. And I'll talk about what exactly drug synergy is, but there are lots of applicable uses of this platform. So you may have an interest in drug repurposing. So many diseases, cancer, various infectious diseases are already treated by combining multiple drugs and giving those to a patient at the same time to achieve the best possible efficacy. So you may want to increase efficacy. You also may want to attenuate toxicity. Oftentimes drugs given at very high doses are very toxic. Possibly if you found two drugs that acted very well together, you may be able to lower the dose of both those drugs and therefore reduce toxic side effects. But this platform can also be applied to basic research interests. So if you have an interest in a certain signaling pathway and how that may interact with other signaling pathways within a cell or within some biological system, combining two inhibitors of both of those pathways may give you some insight into how those pathways are interacting with each other and feedback loops. You may also want to explore polypharmacology. So there are many drugs that have multiple mechanisms of action, and those would be polypharmacology drugs. And those, if you were to use those for drug screening, you would combine two drugs together. Maybe one you know the target and the other one you are unaware of the target. Doing a drug combination study may be able to help you understand more about the target of that drug, which is an unknown mechanism of action. So then what do you need to set up this type of platform? And so the basic requirements that we kind of started with are very simple, but require a lot of in-house development and a lot of people with different expertise to get this platform off the ground to the point where it is now. And so the three requirements that are really important for this is, number one, you need to have compounds with a defined mechanistic class or target. So compounds that you have a good idea of what the structure of those compounds are. You need to be able to say that they are very good at targeting these specific proteins or this specific type of interaction within a cell. The second requirement that is important for this type of platform is you have to have a flexible and accurate compound plating technology. And the flexibility aspect is very, very important, as you'll see in the presentation. And then lastly, you need a data analysis platform that can mitigate assay artifacts, which is key for any sort of high throughput screening. But you also want to be able to view the data in an easy way and allow your collaborators to view the data in a way that's meaningful. And also you want a way to rank synergistic combinations and see which ones to move forward with for follow-up screening and additional experimentation. So starting with the first one, compounds with defined mechanistic classes and or targets, we at NCATS have many in-house drug libraries that have been developed, some using commercially available compounds, some with collaborations through pharmaceutical companies where they've supplied compounds to us. But one of our really common drug libraries that we use for our cHTS platform is the MIPE library, and MIPE stands for Mechanism Interrogation Plates. And that is just a name that we applied to this library to say that there's multiple mechanisms on this plate or in this drug library. So there's 1,912 small molecules in this library. All of those compounds are plated at an 11-point dose response curve. And there's a range of investigational and approved compounds in this library. But the real strength of MIPE is that it is fully annotated. And so as you can see, this image is just demonstrating some of the compounds that are in MIPE. For example, this belinostat is an HDAC inhibitor that is currently in phase 2 and 3. And this is one HDAC inhibitor, but there are many HDAC inhibitors within that class that exist. And so what we've done in MIPE is we have put all of the HDAC inhibitors that are commercially available into this library. So there are approximately 20 HDAC inhibitors in MITE. So the mechanisms of action are redundant, because what that allows is that we can screen multiple compounds that are supposed to have the same mechanism or the same drug target, and see how all of those perform in that assay, to kind of get an understanding of whether those mechanisms are really accurate across all of those compounds. And that is a way that we can use this combination platform to really try to understand basic biology and for clinical relevance. So we have an excellent library of small molecules, and that's one of the main requirements. So then let's talk about drug synergy and why exactly, what exactly is synergy. So the definition of drug synergy is actually quite simple. It's A plus B or A minus B, pretty much any mathematical operation of A and B, and is that greater than what you would expect A and B. So a simple math problem is, if A is 7 and B is 3, if you add A plus B, you should get 10. That is what you expect. But if you added 7 plus 3 and got 14, that would be synergy, because that is greater than what you expected. So I know that that seems very simple, but that is on the basis of most formulas to calculate synergy. That is basically what it is. And so one of the challenges with trying to calculate drug synergy is that there are multiple models that exist. And this chart here is just a really a summary of the different models that are available for calculating drug synergy. And there is really very little agreement within the field of pharmacology about which of these models is the best one. The best thing that you can take away from this slide is that each of them have their strengths and weaknesses depending on what it is that you are trying to do with your data, what the goal of your study is. And so in this pros and cons section, you can see that the Lowy model is a dose effect based model for calculating drug synergy. And so what you can see is that even though it is mechanistically well defined, and because it is uses a sigmoid-like structure in its calculation of synergy, it's not robust, because it assumes that both A and B have a similar Emax, a similar hill slope, and are similar. It doesn't account for the variation that could exist between two things, the two drugs that you're combining. Whereas the Bliss model, or which also can be applied to these other formulas, the HSA, gamma, and beta, these are much more complex, and so they are very robust, and therefore can handle situations where the two compounds that you're trying to combine are at different ratios across your experiment, can be highly different activities of the two compounds that you're combining. But the con of that is that it is more noisy, and also introduces more error into the formula used to calculate it. So since synergy is something that there's multiple ways to calculate it, and there is not very good agreement upon how to calculate it, the thing that we do here at NCATS is we basically use all of these models. We give all of the synergy metrics, and people, our collaborators, can pick and choose which ones they would like to use to measure their synergy, and I'll show you how we display that a little later in the presentation. But let's get back to how can we do this in a way that is meaningful and will reveal drug synergy. How can we combine compound A and compound B in a way that will reveal drug synergy? And so what our standard, or what we tried when we were first implementing this platform, is to do a 4x4 matrix block, which is pictured here. And so what you'll see is that compound A is on the x-axis of this block. And so it exists in 3 doses in this 4x4 block at 8 micromolar, 16 micromolar, and 32 micromolar. And then over here, there is always a well that has absolutely nothing in it, or that's your DMSO control well. So compound A is plated in single agent in this bottom column of this matrix block. Compound B is plated in single agent on the right-hand column of this matrix block. And so you can see that it is plated at a completely different dilution and concentration than compound A. So it's 12, 6 micromolar, and 3 micromolar. And so that is how compound B is plated in single agent in this one well. And then the center of this block is the part of the matrix block where you would be able to see synergy if it was present, because in each well there is the combination of the dose that is directly of compound A and B are in each of these wells, and so you can see the mixing colors. Compound A is red, compound B is blue, and so in this block you have various shades of purple and pink because each of those compounds is in different ratios within this 4x4 matrix block. So if you were trying to do this in a systematic way, say we wanted to test multiple compounds all in combination with each other, you can see how this could get to be a really, really large experiment that would require a lot of resources and time for actual compound plating in this format. So just looking at this 4x4 block, if we only wanted to do one of these, we would need 12 dispenses of drug A by itself to make up part of this matrix block. We would need 12 dispenses of drug B to make up the rest of this matrix block, and then we would need one well with just DMSO by itself, which is our control well. So just to make this one matrix block, someone would need to undertake 25 dispenses or 25 pipetting steps. So you can see that on the right there is an equation for how we calculate non-redundant matrices for n number of compounds. So you basically just take n, multiply that by n minus 1, and then divide that by 2, and that gives you the total number of pairwise combinations. So the example I've given you here on this slide is, let's say that your n is 30 compounds. 30 multiplied by 29, divide that by 2, that is 435 combinations. So in order to do 30 compounds in pairwise combination in a 4x4 matrix block, you can multiply 435 by 25 dispenses for each block. That is a very large amount of pipetting and would be very difficult for someone to do by hand, which is why this must be done in an automated fashion. And so that is what I'll talk about now, is how we have automated this system using this new technology in acoustic dispensers. So looking at this second bullet point, we need a flexible and accurate compound plating technology. And so that is where acoustic dispensers come in. They are the ideal tool for this application. And on this slide on the left, I've pictured 2 acoustic dispensers that are widely used in industry, in various academic institutions. And so what these instruments do, it's actually it's amazing technology. What they are able to do is they use an acoustic sound wave to transfer a droplet from a source plate into another plate, a destination plate. And so if you'll see in this kind of cartoon on the right side of the slide, you have a 384 well source plate here that is sitting, there's no lid on it, it's open, and it has compound in each of the wells. Then at the bottom of that, in this little blue area, that is the transporter, and the transporter creates a wave of sound, which is applied to the bottom of that 384 well plate. That wave of sound hits a very small area in a single well, and that sound transfer takes a droplet, which is shown here in this small blue dot, and it transfers it straight upwards into a 1536 well destination plate. And that is how you achieve the matrix block, or the 4x4 matrix block that I've shown here. And that on your destination plate, you would program your acoustic dispenser to transfer compound A from a certain well into all of the wells that will contain compound A. which is here as I've shown in red. And then you would program your acoustic dispenser to transfer all of compound B into the wells where it should be, as I've just highlighted in red. And at the end of that, you also have your acoustic dispenser transfer DMSO into the corner well so that you have a control within your matrix block. And that is how you achieve this platform. And so you can see that the flexibility in acoustic dispensing is great, because you can literally tell it to take any well from your source plate and put it into any well on your destination plate. This is highly different from a pintle transfer or some other handler that uses tips, because typically any transfer that uses tips, it's going to be static, so you can only go from one location into the same location on your two plates, your source plate and your destination plate. But this is highly flexible, so you can tell it to go from one well of your source to another well of your destination. You can also have it go do multiple dispenses from a single well. You can have it do one dispense from every well into one well of your destination plate. The possibilities really are endless. So just a brief compare and contrast of the two main acoustic dispensers, or the two most common ones. EDC Biosystems makes the ATS 100. LabSite Echo has 3 different models of acoustic dispensers: the 525, the 550, or the 555. And so this is just a brief compare and contrast of both. They both can dispense various sizes of droplets, and they both can also dispense liquids of various viscosity. So if your compound is suspended in something that's not DMSO, let's say it's suspended in water or glycerol or something like that, the acoustic energy required to transfer something in DMSO is different than needed to transfer a liquid that is of a different viscosity. And so both of these instruments can do various liquid viscosity. However, the EDC Biosystems requires a calibration of each of the liquid types that you would like to plate, whereas the LabSite Echo, that calibration is included in their software, and so there is no calibration required. You just specify which liquid you're going to be dispensing dispensing with, and that's all that's needed. The other difference between the EDC Biosystems ATS 100 and the LabSite Echo is that EDC can use any source and any destination plate that you would like to use. You can use any brand, any material. There are no limitations on that. LabSite Echo has chosen to restrict the source plate type that you are able to use. So you have to get your source plates from LabSite Echo. So that does increase your consumables costs, but they argue that their production of the plates is more consistent than you would get if you're using source plates from lots of different companies. And then lastly, there's really big software difference between these two instruments. So the EDC Biosystems ATS 100, the supplied software is very bare bones. There is not a lot of, it's not very user-friendly, and it also does not have full capabilities for setting up matrix screening. It can do some combination screening, but it is not as advanced as the LabSite Echo software is. Also the LabSite Echo software is very user-friendly, and so it was built to be used as a walk-up instrument that a person who has no experience in acoustic dispensing could walk up, create a protocol on their software, and do acoustic dispensing in their plate and have it, and get all of the everything is included in their software. And so one of my supervisors here kind of described the EDC Biosystems ATS 100 and the LabSite Echo as the Android and the Apple of the acoustic dispensing world, because the LabSite Echo, even though it is quite user-friendly, it is very limited in what you can do with it. Whereas the ATS 100, the software is more bare bones, but that does allow for you to do your own customization on top of what they've given you. So those are just two this is just a general comparison of the two. And additionally, the LabSite Echo is more expensive than the ATS 100. depending on which model you need for your needs. So one of the challenges that we initially had upon starting our cHTS platform is that usually what we get from biologists before setting up an experiment is a form like you see in the lower right corner. We get a list of compounds, and the biologist says, I would like for all of these compounds to be combined with all of the compounds on this list. So all pairwise combinations, and these are the concentrations I want, and this is the dilution curve that I want. So that's typically what we're handed. And then one of the challenge with that is that we basically need to take these compounds, put them on a 384 well source plate in a format that is able to plate this experiment as they want. And we also need a map generated for this source plate. The other thing that we need is a reformatter, or a way to actually take that 384 well plate and say, okay, after my experiment is done, when I've done this plating, what is my 1536 well plate going to look like? Where are the compounds at? Where is compound A been plated? Where is compound B been plated? And how is it arranged on the plate so that our bioinformaticians can actually do their data analysis? And the last really critical part of this is shown in the right, is that we needed a program that would generate the file that is actually needed to give the acoustic dispenser to actually create this to actually create a file that is a script file. So all acoustic dispensers have a file that has to be run, they have to use in order to run, to know where to plate, where to move, to take from this location on the source plate and move to this location on the destination plate. And so in the lower right corner, this is just a sample script file. And so you have to have a file like this to load onto your acoustic dispenser so that it knows what to do. And so we had an amazing software developer in-house. His name is Don Liu, and he actually wrote our first automated plate reformatter. And so what it did is you inputted a list of compounds, it created a map for you for your 384 well source plate, and it also created a 1536 well map for you, and it gave you all of the files needed, the EDC script files, in order to actually run your experiment. So this was a very critical step. Without this piece of software, it would have been very difficult for us to be able to do this type of screen in a large scale, because there's so many files that need to be generated. So that was a very big hurdle we had to cross. So once we had gotten past that, then we had to decide on plating formats. And so what we kind of decided on internally was that we would do 6x6 matrix blocks and 10x10 matrix blocks. So I showed you a 4x4 earlier, and in early experiments, it was shown that that was kind of too few concentrations needed in order to actually have drug synergy be highlighted in a block. And so what you can see in this matrix, the 6x6 matrix block on your right, exact same format, and the red and the black colors are for a cell viability assay. That's our coloring scheme. So anywhere you see black, that means that lots of cells are alive in that well. Anywhere where you see red, that means lots of cells are dead in that well. And this is after sometimes 24, 48, or 72 hours of drug treatment. So a 6x6 matrix block is 5 doses of drug A here on this bottom row, and then 5 doses of drug B in the side column, and then in the middle, just like in the 4x4 matrix block, that is where all the drug combinations are at, and you have your well of DMSO in the corner. So each matrix block takes up 36 wells on a plate. Then as a follow-up format, so basically this is the 6x6 is used for kind of a primary assay, and any compounds that look, or any combinations that look synergistic there, we then move those up to a 10x10. And so what that does is that just makes a much larger matrix block, and that allows us to see if the synergistic window is larger, is larger than what it was in the 6x6. And so the 10x10 is exactly in the same format. So you have your drug A is of 9 doses are on the bottom of this matrix block. On the side is drug B by itself, and then same thing in the middle, you have all of your combinations of drug A and B together, and your DMSO well is in the corner. And so again, and so you can see in both of these blocks the area where some possible drug synergy is happening. For example, if you look at this well right here that I've highlighted in red at 44.6% viability, you can see that in the wells, the single agent wells, both drug A and drug B are really doing almost nothing. There are all the cells are alive, 196% of the cells are alive. Whereas if you combine these two doses, you get about 44% of the cells left alive, which is the beginning of drug synergy. And you can see in this 10 by 10 matrix block, the synergistic window is actually much larger than it is because it is a larger matrix block. So you can see in the red area I've highlighted in the 10 by 10, that is where the drug synergy is really strong in this particular block. So those are the two plating formats that we decided upon. And then we implemented our first robotic system to automate this screening. And so in our first system, this was a long time ago, we had one EDC ATS 100. We had a thermo CRS arm for moving plates between the EDC and a thermo rapid stack, which actually just held the plates that we would be using for this. And then we had an Agilent plate lock and an Agilent V-spin in case we needed to seal any plates or spin any plates in our protocol. And the same software developer that built the automated plate reformatter also built robotic scheduling software in-house so that we could do large-scale screening of multiple plates without having a person there to move plates on and off this platform. So this was our very earliest acoustic transfer robotic system. And so the next major hurdle that we had to do is that system was very successful, but the amount of screening that we were doing, it was getting larger and larger and larger. And so the challenge that we had there is that we had to start creating assays where we had multiple source plates. And so why do we need multiple source plates? Well, let's say that this is a very small list of compounds here in the right corner. There's only about 10 or so of them here. We started to get into situations where people were submitting matrix screening where they wanted to do, one drug in combination with an entire drug library. So they wanted to do one drug in combination with 2,000 drugs. And so that is a very large number of combinations, and all of those 2,000 drugs will not fit on one 384 well source plate. And so what we had to do is figure out how could we format our source plates in a way that would allow us to do these very large screens, but also format them in a way where we could account for we need more volume now because we're doing larger screening. We maybe need a higher concentration because our stock concentration have to be higher to achieve our desired final dose. And so some of the considerations that we had to start thinking about is in creating a source plate is what stock concentration do we need to have in that plate so that we get the desired final concentration? What volume of compound is needed to finish the entire screen? Maybe we're only transferring 10 nanoliter droplets of compound in most of our assays, but if you're doing something where you're, you have 2,000 combinations, you're going to quickly move through a lot of compound. And also, do we need multiple source plates? Will all the compounds fit on a single plate, or do we need to expand that over many source plates? And so to kind of get over that hurdle, again, we moved to automation and software development. And so we had another in-house developer named Tim Mearswa who created our automated source plate generator. And so what this piece of software does is just a screenshot from that software. And so what this software does is whenever we get a list of compounds submitted that says I want to do all pairwise combinations of these 30 compounds, all our compound management staff has to do is input that list of compounds into the box that I've highlighted here. they have to fill out a few parameters: the transfer volume, what's the final assay volume, what is the stock concentration of each of the compounds that are on this list, what is the matrix size, is it a 6x6, is it a 10x10, and then how many replicates of this are we going to need? Is this something where we're screening, 5 cell lines of the exact same combinations? Are we doing multiple assay types? You input all of that into this first screen here. Make sure you fill out your settings. And then from there, this software is amazing because what it does is it produces every single piece of information and every file type that is needed to run that matrix screen. So it makes all of the source plate maps. It makes all of the destination plate maps. It also makes the script files needed for the acoustic dispenser in order to actually plate that experiment. And lastly, we use a Perkin-Elmer-Janus in the compound management lab to actually create the source plates to go from the compounds in a tube to the compounds in 384 well plate. It actually creates the Janus files that are needed to create that source plate. So this software in one shot with minimal input makes everything that is needed in order to make the source plates, make the destination plates, and run the screen in an automated fashion. So with the implementation of this new software, we are now able to do very, very large matrix screens. And so now our software had kind of eclipsed our robotic setup. So recently, in the last 2 years, we have set up a new robotic system. And so this is our current robotic system. So on this system, there are 2 acoustic dispensers. Both of them are, this is EDC1 and this is EDC2. So we have 2 ATS 100s on this platform. And this platform was built for us by High-Res Biosolutions. Also on this platform, there is a plate hotel. It's really hard to see because it's kind of underneath this platform, but it can hold 316 plates. There is a microcentrifuge here in case plate can just be spun down. There is a litter and a de-litter hotel on this platform, so lids can be removed from plates. There is also a plate lock and a plate sealer here in case we need to seal plates during the assay, and there's also a barcode scanner, which allows for us to keep track of all the plates to make sure that the correct plates are, the correct compounds are applied to the correct plates. And the software that operates, the scheduling software that operates this entire robotic system is called Solario, and it's the most recent version is the 3.2 version. And this software is actually relatively user-friendly. And so this is just some of the highlights of the Solario software. One of the things that's great with this one is you can actually do graphical protocol design. So this is just a sample protocol that you can create, and it will show you exactly, what in graphical format, what that protocol, what you've told the robot to do. So in this sample protocol, the robot is going to pick up a source plate. It is going to take the lid off of the source plate, and it is then going to move it to cherry pick. And in our case, cherry pick is the acoustic dispenser. So it's going to move it to the acoustic dispenser. And then the next thing that it's going to do is it's going to take the destination plate and pick it up from the lid hotel. It is going to, this incubate step is just going to wait at one of the platforms until the source plate is in the acoustic dispenser. It is then going to take the lid off of the destination plate and move it to the cherry pick, which again is the acoustic dispenser. And then it is going to put the lid back on the destination plate when it's finished, and it's going to stop, and then it's going to remove the source plate, put the lid back on the source plate, move it back to the lid hotel, and stop. So you can really see, if you set up a new protocol, what exactly you told the robot to do in a graphical way, which is really helpful. The other thing that's great about this is that it has simulation mode. So if you're planning a new experiment and you're not exactly sure how long it's going to take to plate, exactly how long each of the destinations is going to take to finish, this format is really great because you can run it in simulation mode. there is a time bar at the top of this, as you can see, and it will give you, if you started at this time, your experiment should be finished, all the spotting or the plating of the compounds at approximately this time. The other thing that's really helpful here is that in this software, you can specify whether you want your assay to be run to be source-centric, meaning that it will keep it'll keep a destination plate on the acoustic dispenser, and it will go ahead and bring in every source that is needed to make up that one destination one at a time. And then once it's finished with the destination, it will remove it, put in the next one. So that is a destination-centric assay design. And then you can also run things in a source-centric fashion. And source-centric it's the same thing. It's going to leave one source plate on the acoustic dispenser, and it is going to move all the destinations and complete every destination needed to be done with that source. Then it will remove that source and put on a new source and continue. And so depending on what type of experiment you're running, how many source plates you have, being able to toggle between those two options can really help speed of your experiment. And then this is just a screenshot of the how you create orders. And so an order is just literally a protocol similar to what I was showing before in the graphical format. So in this order, we have uploaded a script to plate 76 plates. This is our source plate here. All of our destinations are spread across these columns in the lid hotel. It shows you where all your plates are located at. And so this is one order. And so you can create orders in any way that you like. It's really nice because you can visualize all of them. And then you can also do order scheduling, which is really, really been great because you can link multiple orders so that once, for example, this order that is saying is active right now, that means that order is actually running at the moment. But we have scheduled this to say that as soon as this order is complete, order 5570, it's going to start the next order, which is 5569. So these two orders are linked, and it will just automatically start the next one. And so what that has allowed for us to do is that, overnight, we can, start acoustic spotting of an order, and it will be finished when we come in the next morning, barring any sort of error that occurs, that may occur for, various reasons. And so that has actually been a really big advance for automating this entire platform. We regularly plate 156 unique destinations that are comprised from 5 different source plates on this robotic system. And so that is how we've adapted this flexible and accurate compound plating technology for our cHTS platform. And then the last major advance that we really put a lot of work into was a data analysis platform. And so I'll talk a little bit about that. And so this is our typical workflow, and we, for the most part, have done cell viability screening, so using cell titer glow from Promega at either 48 or 72 hours after compound and cell addition. And so we start off our workflow with just getting single agent dose responses, and that's really important because we need to know the concentration ranges of the compounds. So once we have a list of active as single agents, we are going to cherry pick, adjust those concentrations, and then run a 6x6 primary screen. And so our format, current format, fits about 35 6x6 matrix blocks on a single 1536 well plate. And then once we have looked at that, done a data analysis, any compounds, combinations that appear to be synergistic, we then are going to move to a 10x10 follow-up screen. And the 10x10, again, is an expanded matrix block, so you can really zero in on where the drug synergy is happening and what concentration range that's occurring. And so this is kind of our typical workflow. And so what are the important considerations before getting started doing a screening platform, a screening workflow in this way? So one of the major things that I think people underappreciate is you really need to think about what is the goal of your drug screen. Do you have a hypothesis that you want to explore? Are you trying to do drug repurposing? what is the goal, because that is really going to dictate what your screening strategy is and how you'll be able to answer the question that you are aiming for. And then other considerations that are important, the cell line or enzyme, the assay that you're using, is it expensive? Is it difficult to obtain, or is it easy to obtain? Because the number of plates that you can run through for a typical drug combination screen can get quite large, depending on the number of compounds that you're using. And so it can get expensive. And so thinking about, do I really need to use this patient-derived cell line that doesn't grow very well for this experiment, or can I try to use maybe a model that is not quite as applicable but is more amenable to this platform? And then also thinking about positive and negative controls for drug synergy. Are there two compounds that in the literature have been shown to combine to get the outcome that you're looking for? If you can include those in your list of compounds that you're using for screening, then that will really help you see if the assay that you're doing is working how you expect, or if it's replicating previously published data. And the really big thing that is also important for setting this up is making sure that your concentration ranges are where they need to be to see drug synergy. And so in the top, there's a 6x6 matrix block that I've shown you that really doesn't have the concentration windows set correctly. So drug A in this 6x6 block, the top concentration is set at 10 nanomolar. And so that seems really low, but this drug is very active. And so you can see that at 10 nanomolar, this compound is pretty much killing every single cell, and then we quickly switch to a curve on the bottom here where we're doing no cell killing. So the concentration range for drug A is a little bit too high, because in an ideal scenario, you really want your two drugs, drug A and B, to have maybe one or two concentrations that are highly active in this way, but you really want at least two concentrations surrounding the IC50, because it's very difficult to see drug synergy when you have one drug that is acting in a very strong manner to cause cell killing. And drug B, if you look on the right of this 6x6 matrix block, you can see that drug B concentration is actually a little bit too low. So it is actually set at a top concentration of 4 micromolar, but at 4 micromolar, we have about 30% of the cells dead. So we only have about one dose in this matrix block that is giving activity, and it's kind of only giving partial activity. We have 4 doses where drug B is really not doing anything at all. And so for this, what this does, if your concentrations are off, it really minimizes the synergistic window for your assay. And so you can see that this on the right is a delta bliss map. And so that is one of those synergy models that I spoke about. And so what it does is any area you see that's in a dark red color, that means that well is very synergistic. And any well you see that is in a whitish color or has a 0 value, that means that drug combination in that well is not synergistic, or possibly additive, but definitely not synergistic. And so you can see that for this 6x6, the matrix block, the combination, there's only really one combination that has really strong synergy, and all the synergy is pretty isolated to this single column, these 2 columns. Whereas if we move down to the 10x10, the concentrations for both these drugs were adjusted. So drug A was adjusted down, so just from 10 nanomolar top concentration to 5 nanomolar, and so you can see that that kind of gave us what I was speaking about earlier. It gave us one concentration where there's a lot of activity, but 2 concentrations kind of in that middle range, and many concentrations without any activity. And then drug B, the concentration was increased to 5 micromolar, and so that also did a really good job. It gave us 2 concentrations with really good activity, but some concentrations in the middle. And so what that did is that really increased the synergistic window here for this experiment. And so now we have a very large area of synergy as compared to this 6x6 where the area is very small. So adjusting your concentrations and picking the correct top concentration is really important to be able to highlight drug synergy. So here's some screening strategy options. You can do one compound versus every single compound in a drug library. And so what that requires is that your library compounds be plated in an acoustic format. And that's a pretty simple thing to do. We just plate the library all at once. We freeze down sets of it. And then once you're ready to do this type of drug versus all screen, you just add your single drug to those plates. You set up your files and proceed. And so the advantages of doing this type of setup is that it's unbiased because you're doing an entire drug library. And it's also really important for testing if you're looking for drug repurposing. Say you have a disease where there's only one known treatment, you could try that known treatment versus everything else and see if you get anything interesting. The disadvantage of this is that because your library compounds are usually pre-plated, your window, your synergistic window may be off, just like in the concentration considerations I was just showing you. If this particular cell line or assay may be more or less sensitive to those library compounds, you may miss your ability to see synergy. So that's one option. Another option is what we call an all versus all. So you basically just pick in number of compounds, do all pairwise combination. This is a limited list. You're not going to get the entire drug library, but if you have a very focused idea about what you're looking for, then you should be able to find it here by picking compounds that are focused on that hypothesis and doing all the combinations. So that is kind of an advantage and disadvantages of doing an all versus all screen. So then how do we QC this type of data? So you can imagine that this is a heat map of a 1536 well destination plate, which has been run on our platform. And so you can see that this is for a 10 by 10 matrix box. In this blue box, you have, two different matrix blocks. And so typically, if you're doing a cHTS, you would look at the Z prime of this plate. You would look at the signal to background ratio. There are lots of metrics you can assess. But the problem with doing that type of analysis on a matrix plate is that you are going to have a range of activity within this single matrix block. So there isn't going to be anything consistent or shouldn't be across this entire plate because every matrix block is different. You have one part of the block with a single drug in it and other parts of the blocks with two drugs. So it makes it difficult to do a typical QC analysis. Another aspect of matrix screening that's different than single agent screening is there's multiple liquid dispensing. So you're dispensing your cells or your enzyme, plus you're dispensing your assay or your compounds using the acoustic dispenser. And also the scale here is quite large. As I spoke about, you can see that if you just do a small number of compounds, that really is going to increase the amount of concentration of combinations that you're doing a large number of plates over time. The other thing that is important is that the formulas to calculate synergy are not very good at reducing the error that you may get in that particular matrix block. So for example, in this expected formula here, your drug A had 50% response, drug B was 60%, drug A plus B got 30%. The synergy formula or the delta bliss formula here, that's just a change in that bliss model, is basically going to be 0 because, and so that means that this was not synergistic. But if you add just a 10% drift on that exact same formula as shown above, you see that, drug A 50%, drug B 60%, but as I said, you just add 10% drift. So drug A and B, you added its 40% cell viability as opposed to 30 in the above equation. You can see that you would actually get a value here that makes it seem like that's a synergistic combination. So just 10% variance can really affect your ability to quantitate synergy. So how we chose to get around that, one of our bioinformaticians here, Raj Guha and Wu Chen, they both came up with this crowdsourcing QC metric for matrix plates. So what they did is they basically sent an online survey to internal users here at NCATS that do a lot of matrix screening. And just as is shown on this slide, this survey asked people to rate the quality of this matrix block. Not necessarily whether this block was synergistic or not, but just, is this a block that when you look at it, you can think you can trust this data? And we were supposed to rate that as good, medium, or bad. And so what Lou found upon analyzing all the survey answers, he compared the survey answers, good, medium, or bad, to the Z prime of each of the plates that he had taken those matrix blocks from. And so the general rule of thumb, if you have a Z prime over 0.5, then that is a good assay. The closer to 0.8, the better. But what Lou found is that if he actually compared the bad, medium, or good responses from that survey to the Z prime, there was really no correlation between bad, medium, and good matrix blocks compared to Z prime of those plates. So you can see that plates with 0.8 Z prime or close to 0.8 Z prime seem to have really good matrix blocks on them, but they also had some bad matrix blocks on them. And these plates here around 0.4, 0.5 Z prime had lots of bad matrix blocks, but also had some good ones on them. So using Z prime alone is insufficient to indicate the quality of each individual matrix block. So what we implemented or created was this algorithm called MQC, or matrix QC. And that algorithm is more complicated than I think I can accurately explain. But what it does on basic principle is it assesses 5 principles or 5 characteristics of each individual matrix block. It looks at the block control. So the block control is that corner block, that DMSO block. So if you look at this image, the corner DMSO block is right here in this corner. That should be around 100%. There should be no cell killing there. I would not trust this matrix block because for whatever reason, this block has lots of cell killing in the DMSO well. So this block should have bad QC. Smoothness of the block is also assessed. So and randomness as well. So you can see that both these blocks kind of have very spotty signal that doesn't seem to be a pattern to the viability. And so that is something that we want to make sure to count as either a medium or a bad block. So we don't want to trust the data from that. It also looks at the variance of the activity and the monotonicity and applied that to a machine learning platform. And so now what we have is a ranking system where we rate blocks as good, medium, and bad. And so bad blocks don't have trustable synergy, good blocks have trustable synergy. And so what we found is that when we applied MQC to the same matrix blocks that were in that survey and also compared those to the Z prime, what Lou found is that blocks that have greater than Z prime greater than 0.5 have about 90% of the blocks are ranked as good and medium, and about 60% of the blocks are ranked as good only. And so that is a good indicator that if you combine Z prime and the individual MQC metric, then you're going to get matrix blocks that you can trust the data and make sure that you can accurately judge synergy. And then the last large development we had to do was create an online interface for actually viewing this data. And so the matrix client is the online website. The URL is listed here, and so you can go to it at any time. All of the published studies that we have done are there and publicly available to look at the matrix blocks for those screens. And so what's nice is you can look at each of the matrix blocks. You get some drug information on the mechanism of action, and you also get a block of the synergy models and how the synergy models look within that matrix block. So basically, we now have all of these requirements. We have compounds with defined mechanistic classes, we have flexible and accurate compound plating technology, and we have a good data analysis platform. So how have we applied these the cHTS to solve biological problems. These are just some figures on how much we have done. We've really done a lot with this particular matrix, this combination HTS platform. You can see that we have screened over 450 different cell lines and malaria as well as Ebola. We've also done a Zika virus screen. We have 7 publications currently, and there are many still in development. And so this is ongoing. These numbers will only go up from here. So the very first paper that was published with this platform, it was in PNAS. It's entitled High Throughput Combinatorial Screening Identifies Drugs that Cooperate with Ibrutinib to Kill Activated B-cell-like diffuse large B-cell lymphoma cells. And so ibrutinib is a BTK inhibitor. And so the simple screening technology or the simple strategy we used here was to screen ibrutinib versus all library compounds, which at the time was MIPE 3.0. That was 465 compounds. And what we found from that, as shown in this figure from the paper, is that ibrutinib was very synergistic with lots of PI3 kinase signaling modulators. And so that drug combination was further explored in animal models, and there were other results from this paper that are being used to start clinical trials right now. And so this is one application of translational relevance of this platform. We've also applied this to look for synergistic combinations for malaria. And so this is a figure from a paper that came out in Scientific Reports. There were 3 malarial strains, and they were screened against over 2,300 single agent compounds, and then cherry picks were made from there. And in total, there were over 13,000 combinations tested in these 3 malaria strains. Those combinations were then moved to animal testing. And so the big takeaway from this paper was that the current malarial treatment were very synergistic with PI3 kinase mTOR inhibitors. And so this is just a figure of the log P adjusted value of the compounds in the MIPE library grouped by mechanism. And so you can see that if you group all the PI3 kinase inhibitors together, we've got very good, very significant activity here. And so those were then tested in actual animals that had been affected with malaria to see if they were better than the standard of care. And so you can see in this Kaplan-Meier curve on the right, this is looking at parasitemia on the left and then animal survival on the right, that this compound NVP BGT226, that's a dual PI3 kinase mTOR inhibitor that was selected for this study, had excellent survival compared to single agent antimalarials, but was not quite as good as the standard of care altogether. But if you removed one of the compounds from the standard of care and really looked at and combined that with the PI3 kinase inhibitor, that is exactly where you got animals that were long surviving long after they had been infected with malaria. So that is something that's also being investigated in clinical trials now. And then lastly, this is a study that I was involved in looking at a basic research hypothesis. And so it's known that BRD4, which is this protein shown here, it phosphorylates the CTD, the tail of RNA polymerase, and allows for transcription to be activated. And so it was hypothesized that RNA polymerase is a direct regulator of topoisomerase 1, which is shown right here. And so topoisomerase 1 is really important for uncoiling the DNA to allow for polymerase to assess the DNA that's there and start transcription. And so that was the hypothesis, that RNA polymerase was actually directly regulating topoisomerase 1. So based on that hypothesis, we thought that if you inhibit BRD4 and topoisomerase in combination with each other, that should cause cell death, because that should prevent the activation of RNA polymerase and prevent the start of transcription. And so that's exactly what was done in this very simple study. JQ1, which is shown here, is a known BRD4 inhibitor, and camptothecin, which is shown here, is a known topoisomerase 1 inhibitor. And so both of those were combined together. And so the cell line that was used is just HGC116. It's the colon cancer line. And so you can see in this bottom delta bliss map that when you combine BRD4 and camptothecin, you have a very large synergistic window. Whereas all of the activity of BRD4 and on RNA polymerase was shown to be involved with exon 4 of topoisomerase. So again, they created a cell line that eliminated exon 4 from the HGC116 cell line to see if that would remove the synergy. And so that's what's been screened here on the right. And so you can see that if you have a cell line with no exon 4 of topoisomerase, that synergistic window, which was previously there, is much reduced and in some cases antagonistic at certain doses. And so that is a basic research application of this combination HTS platform. And so I'll end by just saying that team science is synergistic. And so this is a picture of everyone in the group from a couple of years ago. There are lots of people on this slide. The people that are highlighted are really the key people who really had the idea to have this platform exist and actually implemented it in a way to get us to the point where we are now. So Craig Thomas is the leader of one of the chemistry technologies group here. And he's the one that actually came to compound management multiple years ago and said, hey, I really want to make this. And the this that he gave compound management was that 4x4 matrix block I showed you in those early slides. And so the person who he handed that to was this person here, Paul Shin, who is head of compound management. And Paul basically worked with all of the other people here to kind of make this platform. Paul is the one that really implemented that very first robotic setup. He couldn't have implemented that robotic setup without this person here, Dongbo Liu, who is the software developer who made that first plate reformatter and made sure that we had a scheduling software to actually do things on that very early robot. Raj Guha is the one who made the online matrix client that allows collaborators to view the data, that allows us to look at the analysis. And all the other people here that are in bold have had really important roles in the implementation of this platform. And without them, we would not be in the state that we're at today. And so that is the end of my presentation, and I'll take any questions.
Thank you, Dr. Wilson, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are the advantages of acoustic dispensing over traditional liquid handlers?
Advantages. I think two of the main advantages of acoustic dispensing over traditional liquid handlers. One is that it reduces the need for consumables. So most traditional liquid handlers, small volume or large volume, involve some sort of tip, which picks up the compound and moves it from one location to the other. And so as you can imagine, the more plating you do, the more tips you run through, so that can get expensive. The other thing that's great about acoustic dispensing is that you are able to use much less compound than you would typically for over a typical liquid handler that does physical transfer of a compound, because tips usually have to go into a certain volume in order to get all the way to the bottom of that well. You have to have a certain amount in the well. Whereas with acoustic dispensing, you can put as much compound in the well as you need to plate. The dead volume on the plates that we usually use is about 1.5 microliters. So you could essentially put 2 microliters in a plate and dispense multiple 10 nanoliter droplets from that one well into whatever destination you would like to, and you don't need to use anymore. So it allows you to really reduce the amount of compound that you're using for particular experiments over a traditional liquid handler that uses chips.
Are there limitations of automated screening using robotic systems?
Yeah, so some of the limitations or challenges that we still continue to have in our cHTS platform is mainly related to software. So, for example, the scheduling software that I showed you on the scenario has a limitation that you cannot reuse a source plate or any plate that is in the same location while another order is running that needs that same plate. So you kind of have to do a little bit of pre-planning about your particular experiment and how it's running to make sure that you don't run into a deadlock situation, because what will happen is if you have one order running, let's say it goes to reach for a plate, and then there's a second order running that at some point needs that same plate. It doesn't have to be at exactly the same time, but what will happen is you'll reach a deadlock where the robot will not be able to pick up that plate that it needs. And so that is something that you just have to think about whenever you're designing an experiment. And so we've come up with a workaround for that, and that is our workaround, and that we just try to plan ahead for situations where we need to have the same plate being the same plate in the same location being used on both EDCs. We just try to schedule things to where they will not overlap. So that there are software limitations, and obviously any software has a limitation in that the people that developed it can't think of every single use and scenario that will be for that particular software. And so you don't realize there's a problem until there's a problem, and then you have to kind of think of a way around it or alter your experiment. So those are just some of the kind of basic limitations.
Okay, it looks like we have time for one more question. Can high content assays be used for cHTS?
Yes, absolutely. High content assays or any type of assays, we're using cell viability assays primarily, and for the most part, they're cell titer glow assays. But I have done imaging experiments using our NCEL 2000. I've also done imaging experiments using the Acumen that we have on our robotic platform. So this is not at all limited to luminescent assays or assays with a basic readout. You can absolutely use this for high content experiments. So don't think that there are those types of limitations. You really can apply this technology to lots of different types of assays.
I would like to once again thank Dr. Kelli Wilson for her presentation. I would also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 24, 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share the announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again. Goodbye.